Hi everyone, and thank you very much for joining us today. Thank you very much for the invite to talk at this really prestigious and really exciting conference. The first thing I'd like to do is to express two levels of thanks. First of all, to you as supporters of the Macular Society for enabling the types of research that I'm going to talk to you about today. And secondly, to the Macular Society for supporting us on all different aspects of this research, not only from funding, but also being able to modified plans to be able to ebb and flow with the world of research. And it's really fantastic to have such a supportive charity that enables us to do this sort of work. So I'm Jamie Ellingford. I'm a research fellow at the University of Manchester, but I also wear a second hat. And so I've recently taken on a role at Genomics England as the lead scientist for genome data scientist, in particular for rare disease. And so I wear two hats in my day job, and today I'm going to talk to you more from that kind of academic, research-orientated stance. All the views and ideas that I talk about are certainly don't reflect the organisation; they reflect my views and opinions. But also, I can give you a little bit of insight into how genomic medicine is happening here in real life in the UK through the genomic healthcare service that is now set up for individuals with ophthalmic conditions. And so a huge amount of that work happens through genome laboratory hubs that are set up here in the UK, where my research group is based here at the Manchester Centre for Genomic Medicine in the Northwest Genomic Laboratory Hub. And we work really closely with collaborators at the University of Manchester, but also with clinicians, genetic counsellors, clinical scientists within the Manchester NHS Foundation Trust. So that was a bit of spiel about who I am. And I guess to summarise that, I come from multiple different angles and hopefully can give a little bit of an overview of where this research sits in the wider picture, as well as some of the specifics of what we'd like to do within this 3-year funded programme from the Macular Society. So the project that I'm going to talk about today was funded in Q3 last year and is funded for 3 years. The title of it is the Manchester Eye Tissue Repository Genome Transcriptome Project. So it's got a nice acronym, METRGT. And the aim of this project really is to understand, firstly, a little bit more about how our genome, how the DNA that shapes who we are, how variation in our DNA changes the way that particular genes are expressed or switched on in different parts of the eye. And as a result of that knowledge, what we hope to do is understand a bit more about how the whole picture of someone's genome, their genomic background, as we call it, influences the way in which certain genetic diseases present. And so we're going to focus in on juvenile macular dystrophies, which are inherited conditions that are caused by single or a small number of genetic changes that influence single genes and can cause really severe consequences to vision, particularly in early life. And so those kind of two dual aims, first of all, understand a little bit more about the basic biology of the eye and how it functions normally. And secondly, how we can integrate that knowledge into understanding a little bit more about how conditions present is really what ties this the different partners in this grant together. And so we have individuals both from the clinical side, from the university side, and we have partners both in places like Genomics England, where we're helping to deliver and shape how genomic medicine happens here in the UK. Pause for a second, because that's a lot of information to digest, and just take a step back and think about kind of the major challenges and the major things that we're trying to address with this particular research project. So one of the strange but increasingly common phenomenons that we see in clinical ophthalmology is that, in particular for juvenile macular dystrophies, is that individuals with exactly the same genetic change may present their condition very, very differently. And so a really extreme example of that is that within a family, individuals may have exactly the same genetic changes, but one individual is completely unaffected and the other is affected with severe visual loss. And what we don't understand as a community is exactly why that happens. And so this research has taken a very specific hypothesis to understand how particular genetic changes, in addition to the one that we're focusing on usually in a genetic clinic, and so usually these variants are rare, they're either completely novel to this family or extremely rare in the population. But how this, in combination with a wider genomic background, may influence and dictate whether the severe blindness actually occurs. And so the group of conditions that we're going to focus on, as I've mentioned already, are the juvenile macular dystrophies. And these are an immensely diverse and heterogeneous group of conditions, both genetically and clinically. And what I mean by that is that clinically they may present very differently. Some individuals may be blind from birth, and that blindness may never get any more severe. Whereas others may start to present with blindness slightly later in life, in their first 5 to 10, 15 years or so. And that vision may get slightly worse over time. Other conditions may be syndromic and involve other organs. So, for example, a certain set of conditions known as ciliopathies may affect both the kidneys and the eye, and in some cases also affect hearing. And so this really diverse set of conditions is underpinned by lots of different genetic bases. And what we mean by that is that there's lots of different genes that may harbor genetic changes which disrupt their function and therefore give rise to these conditions. And the exact conditions that these genetic changes may cause can be overlapping between the different genes. And so we know of hundreds of different genes that can cause rare genetic eye disease. And a subset of those will also cause the eye diseases that we're going to look at, which tend to start presentation during early life. And so that kind of key question that we're trying to address, why does one individual present with the disease and another not? When they have these same genetic changes, we don't fully understand, but we know that there's certain factors which may influence that. So one of those is almost certainly the environment and the type of environment that we're exposed to. But the key thing that we're trying to address here is how other genetic changes may influence that presentation. And in particular, genetic changes that are responsible for the way that genes are switched on and to what extent they're switched on in the eye. And so the grant that we've been given really generously for the Macular Society will help us answer those key questions. And so I'm going to try and give you some of the details, hopefully in an understandable way. But for those of you that are hot on this topic, hopefully give you something also to whet your teeth with and to whet your appetite with, sorry. And so what we're going to do is generate a paired genome transcriptome resource. So what that means is we're going to generate a genome for a certain set of individuals. And for those same individuals, we're also going to try and characterize which genes are being switched on and how switched on they are. So we call this gene expression. And to do that, we generate these huge genomic data sets, which enable us to perform some computational analysis and work out exactly which genes are being switched on for the particular sample that we've taken. And one of the keys to this particular research project is being able to access really valuable and unique resources. And that's where the Manchester Eye Tissue Repository really comes to the forefront. So this was a project that was set up several years ago by our colleagues in the University of Manchester, Professor Paul Bishop and Professor Simon Clark. And they collected tissue samples from individuals that are very generously donated tissue samples from their eye post-mortem. So their eye was being donated for a corneal transplant, and other parts of the eye were being donated to the Manchester Eye Tissue Repository. And this is all stored in the biobank here at Manchester. And what we've been able to do as part of this project is access these really valuable tissue resources, which have been frozen in a minus 80 degrees freezer, so that's really incredibly cold, and keeps these samples really integral, intact. And whilst it's been sat there for a couple of years, has enabled us to extract the relevant material from these samples. So in particular, being able to extract a molecule called RNA, which is the indicator of whether a gene is being switched on or not, and from other resources collected from these patients also to extract DNA. So I wanted to give you a bit of an update of where we've got up to with this with the processing of data and processing of the samples through this project. But first of all, I'd like to give a bit of background of the specific data sets that we're going to generate. So I've said that we're going to generate a paired genome and transcriptome resource, the METR-GT. And what we're going to actually try to do is understand how genes are expressed in different parts of the eye. And the two parts of the eye that we're going to focus on are the neurosensory retina. And so this is the part of the eye that involves, that contains neuronal cells, including things like photoreceptors that I'm sure you've heard of, and are responsible for turning light energy into electrical signals that are transduced through to the brain and allow us to see. And we're also going to generate data for the retinal pigment epithelium, and this is a supportive layer of the retina which helps the photoreceptors and other neuronal cells to perform their function. It plays multiple different roles, including an absorption sink for molecules that are created during this process, as well as providing a pigmented layer that protects other parts of the eye. So we're going to generate RNA-seq, which is a high throughput experiment to characterize every gene that's being expressed in two parts of the eye, the neurosensory retina and the retinal pigment epithelium, or the RPE. And we're going to pair that with a genome. And so in order to generate this data, we first of all need to process these samples that have been stored in the -80 degrees freezer. And so there's some videos that we've recorded for the Macular Society that hopefully we can make available after this talk that go through some of the details of how we do this in the laboratory specifically, as well as showing you some of the equipment we use and some of the fantastic staff that are involved in that process. But in order to extract this material, we need specialized equipment, including hoods where we can process these samples really safely, as well as things like centrifuges that spin samples really, really quickly in order to separate different material within these samples. And our objective here is, first of all, to isolate this really special molecule, RNA, from these two parts of the eye. And secondly, from other samples that we've collected from these individuals to extract DNA. And from this, we create libraries, or we process the data in order to cascade this down the different stages that we need to in order to get data that we can work with. And a key part of that aspect is being able to quality control at different stages to understand exactly how much material we've extracted that we're interested in. And secondly, to understand that that material is integral and intact. So RNA, whilst being a really special molecule, is a marker of genes being expressed, and it's part of this central dogma of RNA, of molecular biology, where DNA is turned into this molecule called RNA, which is then subsequently turned into protein. And so it's an active part of the cell, which tells us, gives us a marker as to actually which proteins are being expressed and which proteins are being used in those particular cell types. So after extracting the RNA, one of the things that we need to do is quality control and understand how much is there and whether it's intact or not. And so we use things called electrophoresis, which is running these samples through a gel using electricity to both monitor how intact the molecules are. We expect to see markers which show us whether a sample is degraded, whether the RNA has been destroyed, or whether it's intact. And we can also start to quantify, to work out how much RNA we've extracted from that particular piece of material. And perhaps the major update that I've got today on our project is that we've been able to extract RNA for way over what the number of samples that we'd expect to be able to generate sequencing data for. And most of these samples are of very high quality, and we have a huge amount of RNA that we can use, which is what we'd expected, but it's always nice to be able to see this as well. And so a first kind of major conclusion of the first year of our project is that the sample sets that we're using are of high quality, and that's enabled us to start to generate these really high throughput sequencing data sets, which are quite expensive, but provide us with an immensely powerful resource to ask the types of questions that we've already talked about. So alongside this, we've also been processing DNA, and from the DNA, we want to generate a genome for these individuals. And a key aspect of this is being able to characterize all the different types of genomic variations that are present in the DNA of these individuals that we've generated the RNA data sets for as well. As we said before, a paired genome and transcriptome resource. And so genetic variation can come in all sorts of different flavors. As you can imagine, some of these variants are really common in the population, and some types of variation can be more common in groups with particular genetic ancestry versus others. And some variants are really rare, or it could even be unique to that individual. So our genome is over 3 billion nucleotides, or individual letters, as we like to refer to them, large. So there's over 3 billion A's, T's, C's, or G's that comprise our genome. And if we were to compare the genome of any two individuals, we'd find millions of differences between them. And so there's a huge amount of data that we need to generate in order to be able to completely characterize for each individual the types of genetic variation that they hold. And secondly, to be able to analyze and interpret what those variants mean. And so there's a whole series of computational steps that need to be there, and I'm going to kind of zoom through lots of these right to the end, so hopefully give you an idea of the types of analysis that we'd like to do once this resource has been complete. But before we do that, I really want to give you an update of where we're up to in this part of the process of generating that sequencing data, that high throughput data sets, both for DNA and RNA, that enables us to ask those sorts of questions. So we've extracted all of the RNA, all of the DNA, and quality controlled all of that for way over 250, approaching 300 individuals. We have enough funds to hopefully generate these DNA and RNA resources for up to 200 individuals. And so we've been able to select the best quality from this resource in order to generate these data sets. And to date, we've been able to use a couple of different techniques and a couple of different providers to generate these data resources. And we're able to have now fantastically been able to select the key provider for the rest of the project. And we've generated the paired resource for 60 individuals already. So that includes RNA from both the neurosensory retina, RNA from the retinal pigment epithelium, as well as a complete genome from their DNA. And so for these 60 individuals, we've been able to understand a little bit about the quality of the data that we've generated, and we can measure that again through multiple different ways. And I'm happy to report that so far that data looks fantastic, and it looks of high quality. And one of the key deliverables from this grant is to release this data to the community, both in a relatively raw form, so that people can use the data to address their own specific research questions, but also in a more processed form to enable anybody to potentially go and or anybody with the research interest in this area to go and use this data, even if they don't have expert computational biologists in their team. And so that's fantastic news. We've been able to take 60 individuals all the way through the process and started to understand about the different computational pipelines that we would apply to this data and how the data will look at the end. And so to go into a little bit of specifics about the types of questions that we're going to ask. So I've told you that the general question is, how does genomic background influence the way that these genetic juvenile macular dystrophies present? But the specific questions that we're going to ask are, number one, which genes are driving, which genetic changes are driving changes in the way that genes are switched on? Number two, I'm not going to go into any detail, but to understand a little bit around how those genetic changes also influence a specific process called splicing. So this is a process of cutting up and sticking back together the molecule before RNA. And so we call it pre-messenger RNA. It involves things that don't need to be there for the mature RNA molecule to make protein. And so there's a process here called splicing, which cuts out the relevant parts of these molecules and glues the other parts together. And so there are genetic changes which also influence that process. We're going to detect those. And for the first time, we have this ability, because we've generated a genome, to identify rare genetic changes that are really driving huge changes in the way that genes are switched on or off. And so for those first two questions, we're looking for more subtle changes in the way that these genes are switched on or off in the retina or in the RPE. But in that third question, we're really looking for these rare variants that are driving changes that substantially change the way that these genes are switched on. And this will be the first cohort that we've been able to do this for, as far as we understand, ever. Having generated this knowledge, we will generate a list of variants that we think are, or we know are, influencing expression in a normal functioning retina. We can then integrate that into how we deliver genomics services in healthcare. So understanding combinations of variants and whether they're present in individuals who present with disease or don't. So to give you a really simplified example of this, we could have a young child that presents with a juvenile macular dystrophy that contains a variant, which is their genome contains a variant which is completely unique to their family, but is also carried by the father. The father hasn't presented with disease, he's way past the age where we'd expect him to present with this particular condition, but his young son has. And so going back to this concept of how a single genetic variant can express itself in different ways, we hope to layer in this additional piece of information. So taking these genetic changes which influence gene expression, that influence splicing, the rare changes which really drive huge changes in the way that these genes are expressed, and understanding whether the presence of these is different between the father and the son. And the reason that can happen is because the son himself could generate his own. During the whole process, we generate things called de novo, or new genetic variants, which are completely unique to us. Or also he's inherited 50% of his DNA from his mother. And so it may be that the combination of the genomic background from his mother, as well as this specific genetic driver of his juvenile macular dystrophy, is what causes the condition to present in the son, but not in the father. And so we hope to be able to be in a much more informed position as to how that works by the end of this project. And there's several deadlines in between where, first of all, we can release this kind of knowledge of the basic biology of the retina, how these genetic changes influence the way that genes are switched on. And then as a secondary objective, being able to integrate this into healthcare to understand more about how combinations of changes can drive particular genetic disorders. A third angle that I hope to be able to come back and talk to you about in maybe 3 to 4 years is how we transition this knowledge into developing treatments. And so if we can understand the complete genetic architecture of these conditions and have a comprehensive understanding of the genetic changes that contribute towards these conditions, that may open up novel angles for us to develop treatments that potentially change the way that genes are expressed in certain parts of the body. And so I think that will be a really exciting opportunity over the next few years, and hopefully something that will be a legacy of the resource that we can release through this project. So I'm going to close up now, and hopefully that sparks some questions and we can have further discussion after this presentation. But I'd like to give thanks to all of the individuals that are involved in this process, yourselves as people that support the Macular Society, the Macular Society itself, as well as some of our project collaborators. And just to name a few of those, Professor Graham Black, who's a professor of genomic ophthalmology at the University of Manchester, Dr. Panos Serginiotis, Dr. Kinga Bujakowska, who is an assistant professor of ophthalmology at the Massachusetts Eye and Ear Hospital in Harvard, and Dr. Ayelet Segra, who's a member of the Broad Institute and really helps us to understand the best approaches to analyze the data to detect these variants that are changing gene expression. So as a team, we cover both clinical expertise as well as computational, and as a wider team, also kind of the experimental and wet laboratory techniques that need to be in place to conduct this multidisciplinary research. And so it isn't possible without teams of individuals Of course, that extends to patients, to their families, as well as the supporting charities that enable this type of research. So I'm going to stop there and thank you all very much for your time and for listening to the presentation. Hopefully it sparked some discussion, and look forward to seeing some of your questions.